The monophyletic animal kingdom evolved from a single ancestral protist, which is thought to have been a flagellated protist, similar to the choanoflagellates. The first thing to develop that really separates the groups off was the development of true tissues, and that gave us the eumetazoan clade. And then the next thing to develop that really separates off the groups is the bilateral symmetry. So we see that right here. Most of our animal groups are going to have true tissues, and they're also going to have bilateral symmetry to them. The group that we're going to start off with talking about in this section is the Lophotrochozoa clade, which is up here. And this clade is it's named for some unique structures that many of the members exhibit. So one of those structures is going to be what we call the lophophore. And this lophophore is a ciliated structure that's going to be used for feeding. And then some of them have a trochophore. And the trochophore is a distinctive larval stage, distinctive looking larval stage. So that's where the name comes from. Not all groups are going to display that. And this really does have a lot of different phylum within this clade. So there's 18 different phylum. We're going to talk about 6 of those phyla and the characteristics of those. So as we start to move through the phylums here, the first one that we're going to go through is the Platyhelminthes. This group is the flatworms. Okay, so commonly these would be called flatworms. And if you haven't figured it out, they're going to be very flat and have a worm-like structure to them. So when we say worm really is a generic term. We'll have several different phylums that contain worms. So this is really just the first of them. These are going to include marine species. We'll also have some freshwater species. And we'll also have some terrestrial ones. The terrestrial ones, even though they will be terrestrial, they are still going to be found in damp habitats. They're going to have both free living and also some parasitic species within them. And they definitely do have that bilateral symmetry. So all of the organisms or all of the animals that we're going to talk about from here on out, they do have that bilateral symmetry, which means that they have a top and a bottom, and they also have an anterior and a posterior side to them. So that would apply here to these flatworms. They have flattened bodies, very flattened bodies. They're going to not have a coelom, so there's going to be acoelomate. So there's no fluid-filled cavity inside of these, which means they're going to be solid structures, but they're solid structures that are extremely flat. They are going to be triploblastic. And another thing is that they're going to have a network of tubes that's going to help pull air into them and then also help with the excretion of waste. So these networks are going to be called the protonephridia. And that'll be characteristic of all of the worms that we have in the Platyhelminthes. So if we take a look at one example of them, the free living ones, these are characterized by what we call planaria. Planaria are very common in kind of muddy ponds or retention ponds. And if we look at an example of them right here, notice that they're going to have just one opening, body opening. So they have one cavity that's going to, for the most part here, it's just going to serve as the mouth, because the nephridia, the photonephridia, those are going to allow the waste to kind of diffuse out of them. So this doesn't really have to serve as an anus. In this case, we don't even have an anus. If we take a look at the body, they do have this digestive tube that runs down it. They are going to absorb or eat food with that mouth, which is located about halfway down the body and on the bottom side, so on the ventral side of it. That's going to be called the pharynx. They are going to be very thin, so they just have a lot of things can diffuse into them, so air can diffuse in, the waste can diffuse out. And notice that they are going to have some centralized nerves at the end, which we call ganglia. This is going to give us the beginnings of a brain. It's not a brain yet, but it's a centralized network near the head. And they've got some eyes or eye spots there, very, very simple eyes that really can just sense kind of light and dark. So light intensity. And this ganglia network is going to help them to really organize and start to put together sensory input. There's also going to be some nerve cords that help to extend this network of nerves down the body of the organism. There's also going to be some parasitic species in this phylum. And for the parasitic species, these tend to have a tough outer covering. And this is going to help them survive when they're in their host organisms. So they also have a lot of reproductive organs inside of them. Many of them are just going to be full of reproductive organs, and that'll make up the major part of their bodies. This group that we're seeing on this slide, this is what we call the trematodes. And specifically, this is showing the life cycle of the blood fluke. Blood flukes are very common in third world countries. They are a major cause of disease, right behind malaria, so considered to be the second highest disease caused by some type of animal. And one thing that's unique about a lot of the trematodes is that they require 2 hosts to complete their entire life cycle. So when we're talking about the blood fluke completes part of its life cycle in a snail, so that's going to be the larval stages. And then it needs to infect a human so that it can complete the adult stages and reproduce. So if we don't have both of those different organisms, then we don't get a full life cycle. These have to have quite good, quite well-developed systems of evading host responses, because not only do they have to evade the host responses in the snail, they also have to evade it in the human. One way that they do this is they tend to mimic host proteins so that they're not recognized as something that might be foreign. And so in that case, the immune system doesn't actually attack and destroy them. Another parasitic species is the tapeworm. So that's what we're seeing on this slide. And tapeworms, they can be small, they can actually be very long as well. And they are very, very thin and very, very long. So that's where the tape part comes from. If we look at this picture, they have a really pretty scary structure to them. This would be the head of it that we're seeing here, and the head is called the scolex. But what's unique about these is that notice that it's got all of these little hooks around that head, and those hooks are going to allow it to hook in and really attach very well to the intestinal lining of the host organism. So once they attach to the intestinal lining, it's very hard to remove them because they're really stuck there. We don't have any mouth at all when we're talking about the tapeworms, but since they're so long and so flat, they don't need a mouth. They're just going to absorb all the nutrition directly through that epidermis. So there is no need for a mouth. There's no need for an anus on this one at all. These do have a lot of sexual organs, so reproductive organs. Those are called the proglottids. So those are basically just sacs of reproductive organs, and they're going to make up a lot of the interior of these tapeworms. The next phylum is quite different from the Platyhelminthes, and these aren't worm-like at all. These are called Rotifera. And Rotifera is commonly referred to as the Rotifers, and the name comes from a word that means wheelbearer. We call them wheelbearers because they have this kind of ciliated ring on this one side of their body. Now these are tiny little animals, so very small, and you might mistake them when you look at them first off for a protist because they are so small. But even though they're very small, they are going to be multicellular. So they're multicellular, they have a lot of little organs stuck inside their bodies. These are going to have an alimentary canal. And so what this alimentary canal is this is a digestive tract. So that means that we have 2 openings. So this is the first one that we've seen that has 2 openings. We have a mouth, so we have a place for food to come in, And then we also have an anus for waste to exit from. So we've got all of that inside this little organism, and this is going to have a pseudocoelom. So if we look at these, you can see that there's a lot of different structures inside of the body of this organism. They are known for their jaws. So they have jaws that are called trophi or trophi. These can be used to grind or crush their food. And then another thing that is unique about these is that they do exhibit some parthenogenesis. In parthenogenesis, it's asexual reproduction, but it's asexual reproduction with eggs. So we have eggs that are produced, they don't get fertilized, but they still develop into organisms. So this is fairly unique. There are some other organisms that do it, but this is the only one that we're going to talk about that exhibits this parthenogenesis. This is something that some of the species in this phylum can perform, not all of them. There are some species that will only reproduce sexually. There's others that only reproduce asexually. And there are some groups that have been reproducing asexually for many, many years, so millions of years. And it's unclear how they've been able to come up with a whole lot of genetic diversity to survive so long only with asexual reproduction, but there is evidence that they have been doing that. So asexual reproduction is very common in this specific group of invertebrates. The two phylum that you see on this slide, the Ectoprocta and the Brachiopoda, these are two phylum that do exhibit that lophophore. The lophophore, it is a crown of tentacles. These tentacles, they are used for feeding. They're going to actually pull water in, and then it's going to help to trap food particles inside of it. These are going to also have that alimentary canal. And again, the alimentary canal is a full digestive tract. So we have two openings, we have a mouth, and we also have an anus here. These two groups, there's no head, no distinct head. And they're going to be sessile. Okay, so these are not organisms that are going to be moving around. So they may look, we'll see one group looks very plant-like. The other one's going to look like another invertebrate group, but these do also have a true coelom. So they have that fluid-filled cavity inside that's developed entirely from the mesoderm tissue. If we look at a picture of the first one, so this first picture is the Ectoprocta. Ectoprocta, these are called moss animals sometimes because they look like a moss. So they're very plant-like in their appearance. What we're seeing here, that would be that lophophore. So that's that crown of tentacles that is going to assist them in feeding. These have a hard exoskeleton, which is unique in invertebrates, but we will see some other groups that have some types of skeletons to them. These are very important structures on the coral reefs. If we talk about the other phylum, this is the Brachiopoda. Brachiopoda at first glance may look like a clam structure. These are not clams. Those will be in the phylum Mollusca. So they do have that structure, but notice that they're going to sit flat, where when we look at the clams, they're going to sit upright. So they're going to be opening up rather than opening to the side. These are commonly referred to as the lamp shells. We can see right here that crown tentacles, which is the lophophore, and these are going to actually attach to the seafloor with some type of stalk to them. So with both of these, they are sessile organisms. They are going to attach to a substrate in some way, and we don't have a distinctive head on them like we typically think of when we think of animals.